Welcome everyone to this week's Data Science Seminar Series. I'm David Bader, and I direct the Institute for Data Science at the New Jersey Institute of Technology. This is the final seminar for the season for the fall 2020 semester, and we're very pleased to have Dr. Chandra Bajaj from the University of Texas at Austin speaking on learning to correct form and function with reinforcement. Dr. Chandra Bajaj is the director of the Computational Visualization Center at the Oden Institute for Computational Engineering and Sciences at University of Texas at Austin, and a professor of computer sciences and holds the Computational Applied Mathematics Chair in Visualization. His research interests span the algorithmic and computational mathematics underpinning of image processing, geometric modeling, computer graphics and visualization, structural biology, and bioinformatics. He applies these algorithms to structure, elucidation, and reconstruction of spatially realistic models of molecules, organelles, cells, tissues, and organs from electron microscopy and bioimaging, and fast high-dimensional search and scoring engines for identifying energetically favorable molecular binding conformations, for instance, in virtual screening for antiviral drugs, and integrated approaches to computational modeling, mathematical analysis, and interrogative visualization of the dynamics of electrical signaling and oscillations among neurons in the hippocampus, which is the central area of learning and memory in the human brain. He's a member of the Advisory Committee of the National Institutes of Health, National Center for Research Resources, National Biomedical Computation Resource, and past panel member of the National Academy of Sciences and the Vietnam Education Foundation. He was recently elected chairperson of the National Institutes of Health Molecular Structure and Function Study Section D, and for his research and academic contributions, he was elected a Fellow of the ACM and a Fellow of the American Association for the Advancement of Sciences. If you have questions during the talk and you're on Zoom, please feel free to ask, or we will have a chat session at the end of the talk for Q&A. Thank you, Dr. Bajaj.
Thank you, David. You know, the talk, as you'll see, will be on a little bit of in the context of molecular modeling, but it's also through the lens of machine learning, which has been my passion as for the last maybe now 3 or 4 years. And the deeper I get and the more I learn, the better I feel that this is a thing that's going to allow us to blend all the interdisciplinary sciences that I like to do, which is mathematics, computer science, statistics, and of course, physics and chemistry and biology. So with that, I want to show you a little bit of a video. This was done about 35 years ago using computer graphics animation. It's a video that was developed at Purdue University by Michael Rossman, Tim Baker, and others. And this is of a phage virus, which infects bacteria, and it uses a plunger mechanism to inject its genome. As viruses are parasitic, they can't replicate by themselves, so they have to have a host, organism that has the machinery to replicate. But what you're taking away from this is its exquisite form and function that that it's evolved to learn and develop for its existence. And the existence of the life cycle of these viruses is of keen interest and will always be, because these are organisms that are not alive, but they are chemical organisms that evolve to create reactions that allow it to duplicate itself. Of course, harnessing the machinery of others. And so this is a study that, together with several collaborators who I'll acknowledge at the end. I was embarking at about a decade and a half ago. We were trying to search for a antiviral or possibly a vaccine for HIV, which was all prevalent. And if you just look at the life cycle of an HIV, it uses its envelope proteins called spikes, or the N protein, and it signals the cell, as you're seeing on the right-hand side. And then after a signaling sequence, which involves several different complementary partners, it fuses its envelope, which is made up of what are called bilipids, very similar to the same envelope that encloses a cell, and injects its payload into the cell. And then it doesn't stop there. It's got this wonderful retrofitting machinery. It splices its viral RNA into a DNA, cuts the DNA up, and then splices it into the genome of the host cell. What's virulent about HIV is it infects exactly the immune cells that are responsible to keep us alive, the immune response, which is what vaccines are targeting, that the antibodies created for antigens like little pieces of viruses can be quelled. So it's our humans' response to building antiviral proteins automatically in some sense. And this is the same thing for SARS. But this was 15 years ago, and this is happening now. And so the last 3 years, what we're doing is reinventing the wheel, as it were to say, but being better. So the project that I've embarked on since about now 2.5 years has been, not only do I want to learn how to see what the shape and form of these mechanisms are, but also how do I control them? And once I harness that, I can make things behave the way I'd like to. So I can control them to be better in their task of fighting infection, and also better in their task of achieving a form that would make it function better or achieve its function faster. Now, the tools we have has been steadily improved over time, so we can see further and finer. But when it comes down to the nano world of angstroms and nanometers, 10 to the minus 10, 10 to the minus 9, and even smaller, we resort to the electron microscope, which is more a nanometer resolution. Per pixel spacing is about a few nanometers. But from this, we can image viruses. And you can see from a single snapshot, I can go back and find this virus in multiple different orientations, because it's a in vitro sample. It's in its nativity, and it's moving around. And these are all mature, phages. But from the single snapshot, one can go back and reconstruct a full structural model of this phage, and also learn from multiple methods of clustering and so forth, how many different metastable states that the macromolecule lies in. Now, all this is all computational, and it's been one of the biggest successes in the last decade or so, so that we can, from imaging at the nanometer level, recover shape and form. But what we don't have is a video camera, and especially a video camera which might function at sub-milliseconds to picosecond time frame, or sub-nanosecond time frame, to observe every little interaction and every little action. So what one has done, together with lots of other giants in the field, develop computational pipelines. And this is both for identifying a target from microscopy and saying, I need to inhibit the entry mechanism of this virus. So I want to prevent infection. So I need to know exactly the interacting molecule so then I can design a suitable inhibitor that would interfere with the entry mechanism by binding to it before it can bind to the cell receptor and do its signaling mechanisms. And this protocol is all computational and has been, what we have developed. What I'm going to do in the rest of this talk, I'm going to just pick up one of them, because each one of them is an intertwined set of computations and analyses, all stemming from data. So the goal of this talk is, how do we use data to not only learn form and function, but also how to control it? And that's where reinforcement learning is going to play a role. And what I'm going to target is this little box right here called the target refinement gain. And so what is typically done is, you've got more than just the electron microscope. You can also take individual targets like proteins and attempt to, through the science of crystallography, try to make it into a crystal. Sorry for that. My computer sometimes has a mind of its own and flips slides. But if you imagine that through X-ray diffraction, which I haven't shown you on this very busy slide, you are able to determine exactly the sequence of the protein and also possibly its structure. And as you have determined its structure, you've got an alternative model. And that is actually pretty higher, it's at a finer resolution than the model which you can obtain from something like a cryo-electron microscope. Here, typically, you can get models of the resolution, feature resolution that you can discern, say, to be a nanometer, and with careful, repeated calculations and lots of other data, you can make it down to maybe, 5 angstroms. But with X-ray diffraction, you already can get about 2 or 3 angstrom feature resolution separation. So that way you can see atoms, while here you can see groups of atoms in the other case. So the refinement gain is, if I can, for the same target, if I can give you a higher resolution 3D model, can I align it with the coarser resolution? And hence I can refine the coarser resolution to the finer. And if you may ask, why would you want to do that? Well, in the crystalline form, while you've got higher resolution, the molecule is in a different conformation quite often because it's in a lattice. While with imaging, you're getting it more and more in its nativity. And the nativity conformations are the clues we need to see, is in what form and what shape does it poise before it interacts. And it's also through evolution, just like we have evolved. It has, quote unquote, learned that it conserves its most sensitive binding regions. So the actual pocket where some interaction is going to occur or the docking is going to occur is quite often covered with a flap, and only when necessary the flap opens up. So learning the flexibility of the model becomes a key sub-problem and a problem that is inherent. So this target refinement gain is really a flexible 3D registration. And why do I call it a gain? I'll explain in just a minute. But for the first case, let's understand that a molecule is like soft matter. It's, we are building models and showing pictures as if this was a macroscopic object, but it's really only for our intuition, but not for our sake of getting misled. but it's a dynamical system, which is somewhat in a liquid, like most soluble materials are, and it's more like, you know, again, a macroscopic rough analogy is like an oil droplet in say water, but it not just pure water. Water can be an electrolyte, so it could be in a oil droplet in your favorite Gatorade drink, and that Gatorade drink is very viscous, so it could be an oil droplet in honey. You know? So those kinds of analogies get you closer to the kind of dynamical system which is continuous and constantly changing its state. And what is the state of this dynamical model is given by the configurations it takes and by our mathematical representation of that configuration. So if I was to think, and somehow I've lost my mouse suddenly, but if you were to look at this picture on the lower left side, there's this blobby shape, and that's representing, you know, the density of this molecule um, within its nativity. And the nativity is the white, which is all water with lots of floating salts and ions around it, the electrolytes I was talking about. And the native shape is the true shape, as far as our imaging can tell us. And the crystalline shape is a little bit misfolded. So the game we are interested in, and both of them are the same object, they're just different representations. The game we are trying to play is, can I fold that protein into its native shape? Hence, I can deformably register these two, and hence I can get a more accurate refinement of the molecule. Now, what that means is folding means I'm going to play the game of controlling this molecule. So I want to find the optimal control, which is the minimum, you know, set of moves I should make such that I can take this very complicated object, which we'll see how the state space is going to be very large. And I'm going to do this in a agent, you know, reinforcement agent learning scenario. So as one can also understand that, you know, this problem that we are after is something like an optimization problem. You know, we want to guide the system to a given target state, which is going to be done by an agent, which is going to control the dynamics of this object and make it change and achieve a certain correct form. And I want to use that dynamical system, or I want to control that with a small control, which I'll call f of t, which is on the second side of this. Again, my mouse has disappeared, so I can't point to it. But the dynamical system itself is a spatio-temporal moving blob, which is governed by the equation on the top left. Del u del t, the change of the state, is governed by partial derivatives of space and maybe also environmental external factors, which are given by yt, and the controlling force is f of t. And what you want is to go back and minimize a loss function, but the loss function that we're trying to minimize is the observation loss that measures how well the final state that you achieve approximates the desired target state. It's like, you know, it's the folding game. You know, go back and fold this such that you can fit the exact guide, because the exact guide is our best guess at the configuration this molecule is going to take. And hence, if I folded that necklace or the protein, then I've got at protein resolution or atomistic resolution, a configuration that I want to use for the next step of my computational viral search pipeline. So, you know, I can't give you a very deep background onto when you're representing molecules. You know, what are molecules? You know, molecules are many things, but I I feel that if I start at the beginning, I'm going to take, you know, the entire lecture then. So I'm going to use the often used Latin phrase in medias res. So I have to start in the middle, and starting in the middle, you know, I'm going to abstract out a few things, and then other things that are important I will mention as we go along. So how do molecules achieve their form and how do they function? They're saying molecules show atomic granularity and the existing of covalent bonds that connects atoms into stable machines of defined geometry. Well, this is our mental and, you know, representation of it. Of course, you know, each of these atoms, many of them are highly crucial and many of them are semi-crucial, but there's nature abhors waste. So every atom is important in some sense, but important not in a static sense, that the right time, the right atom, and the right set of objects are going to become important. But it's blessed with its kinetic energy, and so because of the temperature, these atoms are in thermal vibration. And this is causing for random diffusive behavior of this molecule as it moves. But strangely enough, you know, uh, two molecules in a micron-sized cell, which is packed, is imagine it be packed with people, and you're trying to meet your friend who's on the other side of the room. Well, molecules have a way of using crowding behavior to go back and find each other exactly. And viruses do that too. They find their specific host cell that they have to get into. The notions of inertia and gravity are not as dominating as are the notions of internal forces that, and some are long range and some are short range forces. So in this very virulent activity, which is from the typhus molecule, there's an inhibitor that is going and binding to a receptor molecule. The takeaway there is the electrostatic potential. And this is blue. I'll play this video again for just a second. But blue, this is this molecule. But if you were to take the takeaway for that video, you can see the cancellation of the red and blue. That means if red is negative and blue is positive, then red and blue cancel out, which says, oh, these two molecules have a way of lowering the energy by grabbing onto each other. And that is the natural force, which is pretty strong in doing what is called binding affinity between two partners. And that's how molecular recognition, one of the first principles of molecular recognition, works. There are many other terms that we have mathematized. So how do we represent these molecules? We discern the position of every atom. Oh boy. We find masses because we know the type of each atom. It's hydrogen, carbon, oxygen, nitrogen, and so on. We have something called the van der Waals radii. This is where 99.57% of all of the electrons that are part of that atom are orbiting in that radius. They also have partial charges. It's just by electron sharing, and you know, if two atoms which are neutral come together, then they share their electrons like water. Water is like Mickey Mouse. It's got a big oxygen and two hydrogen ears. So hydrogen neutral has, of course, no charge, but once it binds to oxygen, it shares, you know, one of its electrons, its only electron, with oxygen, because oxygen has some free, you know, orbital paths which are stable around it. So it develops a charge of minus minus, and hydrogen develops a charge of plus. And so these partial charges all accumulate to give you a charge per atom or a charge density. And this charge density, coupled with the solvent charge density, which are called mobile, gives you what is called the electrostatic potential. And that's what I'm showing you here, colored in blue. So the electrostatic potential, very much like a tractor beam pulling one molecule, pulling the other. It's not gravity between two large planets that are going to suck a planet to the other. It's this electrostatic potential caused by the bonded nature of these atoms, specific bonds that are created that, that cause for this. Of course, in the nativity, which is water, they have all of these charges. Now, I'm spending time explaining this because I need to understand this mechanism well enough so that I can understand not only its state changes and how it behaves, but what is it that I can control. And I don't have, on the likes of nano tweezers where I can pick up an atom and move it, or like an actuator of a tiny little robot where I can move the joint angle between the bonds. But I can do something indirect. And what I can do indirect is control what I call the Hamiltonian with a small amount of control. So I can change this force field. So to understand that force field, we have to understand a little bit about why these molecules move and how do they move to take definitive shape and how do they change their shape when they interact. It's all governed by something we call the Hamiltonian, the energy that is consisting of two parts. One is the self energy of the system, and the other is the energy from interacting with the environment. And these two terms, there are many models, which I'll show you, are all captured here. Maybe I'll show you that first. So over the last maybe 4, 5, 6 decades, or before even earlier, people have been trying to build models of nature. And one of the things that has taken greatest challenges has been how do you get accurate mathematical models of molecules and understand how they behave, the full dynamical system. So people have built all the way from molecular mechanical, again, my mouse has disappeared, but from molecular mechanical, statistical mechanical, density functional theory, ab initio, quantum calculations. These are all models of what I call the intramolecular energy of a molecule. And I want to control that, right? So I need to understand what model I'm going to by learning. The other energy that comes in is the solvent energy. It's the molecule-solvent interaction. And that is because water is also all molecules, and any ions which are floating around the electrolytes, it could be just salt dissolves into, say, sodium ions and chlorine ions, or potassium ions and chlorine ions, and so on, are all with a certain distribution. So the field theoretic model assumes a Poisson-Boltzmann-like distribution, or a Boltzmann-like distribution, and the Poisson-Boltzmann equation is the electrostatics equation under a Boltzmann-like distribution of ions in the solvent. But you could do even finer density functional calculations for an all atomistic explicit model. And of course, they behave like magnets, like poles, repel each other. So a positive and a negative charge on a bond is like a little dipole. And so there's a magnetic behavior to them too. Instead of going too deep, the essential takeaway is that there's this gigantic Hamiltonian. And this is why molecular dynamics or even molecular protein folding and other things has been a great challenge. And one of the big challenges that defies even the best of supercomputers is that if I'm going to, come up with a model and I'm going to understand and predict the behavior of this model throughout, then I have to just think about the forward problem. The forward problem is, how do I tell you what the next configuration is going to be of this molecule? Well, if you take the derivative of this energy with respect to all the free parameters, which are given by your state parameterization, then that is a force field. So that's a force vector, the partial derivatives. And the forces, both intramolecular and intermolecular, act on each atom. So each atom is then subjected to a force, and that force makes it move under, say, even Newtonian laws, as mass times acceleration is equal to the force. And this is for thousands to tens of thousands to maybe a million atoms, which are making up a virus structure. And they're all, each atom is subjugated to the force. The force is the collective force of the molecule, and also a collective, a force which is gained by the current configuration. So every time you change the configuration, the force field is dynamically changing, and the molecule is subjugated to different forces, and it keeps moving around, right? And we want to control this. But to control this, we first need to understand it and learn from it. And I'm not going to be doing the old way of things. What I'm going to tell you first is how I'm going to learn. And I'll show you these videos later, but I'm going to learn this using this nice reinforcement learning game. This is my target refinement game. So I'm going to think of a molecule solvent as an agent that I'm going to, is going to perform actions. And of course, its actions are going to be controlled by its Hamiltonian in its nativity. But an episode in this game, which is my folding game, is I'm going to take this molecule, and I've got a guide, which I call the map, the cryo-EM 3D map. And I'm going to fold this molecule to fit into this map. And this molecule being a dynamical, system. So this agent has to learn to control properly. And the actions the agent's going to take is going to be by coming up with this E control, this little extra Hamiltonian term that it's going to add to the current Hamiltonian, which is governing the system's dynamics. And this is like taking a very complicated process and saying, I'm going to give you some feedback. But that feedback is not only going to stabilize you, but make you move in the right way. So this feedback control system is what we are after. And we want to learn them. So we are going to learn the forward dynamics, which is this molecular dynamics in this picture. Again, my mouse has disappeared for some reason. But the agent is that pinkish box, which is saying every time it needs to make a change, it's going to change the Hamiltonian, and the molecular dynamics model that it's going to learn is going to move it. It's going to move it, keeping everything consistent. And so the agent plays the game with the molecular dynamics forward simulator. And of course, what we'll get up is you'll see this variational autoencoder that we'll be using in a way to smartly capture, from every trial of controlling and every, configuration the molecule takes, I'm going to learn an embedding space where I can start to make very nice clusters and very nice ways of understanding what the dynamic really is all about. So this is the scenario that I'm going to now go into detail. If there are any questions, feel free to interrupt. This is a good point. Okay. So the first step in this exercise, I'm assuming no questions, David?
I don't see any questions.
Okay. Well, that could mean two things, that I'm not reaching everyone, but I'll go with the other option that let me say something more meaningful before a question comes up. So the first stop of our enterprise is saying, can I learn the molecular dynamics? If I want to pick a model, that was the old way in which we were working. Saying, I've got a lot of understanding of what the essential dynamics can be captured, and from having studied all those models, density functional theory, or simple molecular mechanical models or statistical models. But what I'm going to do is take them as semi-constraints, and I'm trying to find what are the essential moves that are legal moves in this molecular dynamics. So what I'm going to learn is a stabilized, balanced, reduced Hamiltonian for molecular dynamics trajectories. And I want to do this for not all of the range of motions, but for limited ranges of motions. And if I was to think of it computationally, this is exactly what people have been doing with something called principal orthogonal recomposition, PCA, Laplacian eigenmaps, diffusion maps. But what I'm going to tell you about is something which is the Koopman operator and how we can use deep networks for the Koopman operator. So the Koopman operator, the name given is to Bernard Koopman, who together with von Neumann were answering questions of the following type. They're saying, we can see lots of observations of these dynamical phenomena, right? And each time we are looking at this, is saying, what is the underlying linear operator that I can, it could be very high dimensional, it could be infinitely dimensional, this operator called K, which takes a state which is expressed in my, function space F and moves it forward into that function space. So it's like I'm observing a video of this molecule moving, and I'm saying the video is my observational parameter. So this observational function is called G. And if I just look at the G, the observations, so from my G, can I find a mapping of how this dynamical system is changing? So what is the transfer operator that takes a single frame of the video to the next frame of the video? And that transfer operator is given by G of xt plus 1 is equal to G of f of xt. So the G is the observational function, and x is capturing my true representation of the dynamical system that's moving as an unknown function of f. So both f and g are unknown. And can I find a compositional operator k which captures this dynamics, right? So learning the Koopman transfer operator is a data-driven approach to linearly encoding the evolution of the value of an observable time-dependent function. And what we are trying to say is, all I'm going to give you are observations. All I'm going to give you are data that you have sensed, either with your electron microscope or with, and you've taken snapshots of this thing in different stages. Can you find me a linear operator? So if I was to think of this in a continuous setting, what I want to do is, and I'll tell you why it's a reduced dynamical modeling, is I want to find, a representation of this linear matrix. And how do I capture this matrix? I'm going to capture it by its eigenvectors and its eigenvalues. And these eigenvalues and eigenvectors in the continuous setting are eigenfunctions. And these eigenfunctions have to be exactly the eigenfunctions that span the composition of G and F. So I'm trying to look for this matrix K, which operates on G, which behaves exactly as the G composition with F in the time domain. And I can do this for not only scalar observations, I can do this for a whole bunch of vector operations. So the top and the bottom are very similar, except I'm saying that it can be for a whole value observations which are vector values. So G doesn't have to be only a scalar function. And so as an example of a dynamical system, I'll show you more on molecules in a second. But for this is a very well-known fluid dynamical system. It's called the von Karman vortex tree. It, from wingtips of aircrafts and so on, vortices are created, and that's why planes have to fly a certain distance, because that vortex buildup is pretty, turbulent flow. and will ruin the aerodynamics of streamlined flow and lift and drag of any ensuing plane that gets buffeted by these vortices. If I was to look at an approximation, which is what which are called the Koopman modes of the system, then this is saying, here's the first eigenvalue eigenvector, which is the slowest mode of motion. The slowest mode of motion is stationary. It's giving you what is the average stationary field across all the dynamics. The second one is giving me the second dynamical mode. And the third one is giving me the third. And the fourth one is giving me the fourth. And of course, the true dynamics is nothing more than a linear sum of all of these modes. So this linear sum of all of these eigenmodes. So clearly, if we want to build an approximation, we're saying, I don't have to get every little nitty-gritty. And notice that these eigenvalues are complex, right? That means there's not only a translationary motion, but there's also a circulatory motion. Whenever there's a rotational motion, then eigenvalues are complex. Notice also that these lambdas are less than 1. So this is a very stable system. It's also a stationary dynamical system. Because if the lambdas were greater than 1 in their real part, over time, they get amplified, as you can see in this expression. So if it is greater than 1, then these would start becoming bigger and bigger. Right? So, and that can happen in chaotic phenomena. This tornado is building up steam. It's got in that region of this dynamical system, its eigenvalues are complex, but its real part, which adds to also the total amplitude. Of course, the complex part is also important as to the angular rotation that's occurring. The thing is amplifying. In our control systems, we are going to make sure that the approximations we're doing are stabilized so that we won't want to make the eigenvalues to be always having a real part. And this is also well based in theory. These are called Lyapunov stability constraints and also Stein's stability constraints. So what we are going to do is what Koopman was envisioning, and we're going to do this computationally. We're saying, let's think of this complicated dynamics, spatio-temporal dynamics, as modeled by a single matrix, A, which is changing the state. So my state is ut at time t, and this update of the state in a discrete system, discrete digital system, is the state at t plus 1 would be A times the state at t. Very much like a Markov process. And I have some control, and that control is f of t. And this control is unknown. And also the matrix B is unknown. So I've got data, which is various time steps of U, but I have to learn A, I have to learn B, I have to also learn F. And I'm going to do this in a number of ways. Of course, this is not a new problem. And this is a problem that has, in the field of stochastic optimal control or even deterministic optimal control, people have looked at for a long time and have come up with very nice foundational work. One of the simplest is called the convex quadratic optimal control objective, saying, suppose you are minimizing the cost of your control based on a quadratic function. So your cost functional is given by U transpose QU, and also the controlling function itself, ft is ftr ft. So the overall objective is convex and it's quadratic because it's expressed in exactly this form where Q and R are both positive semi-definite matrices. And you're subject to this constraint where ut plus 1 is equal to aut plus bft. And if you want to solve the system, because of the fact that Your objective function is convex and quadratic, has a unique solution, and your constraint is linear. You can solve this analytically using something called the discrete algebraic Riccati equation. Um, so to cut a long story short, I was just looking at, I've spent a little bit more time than I thought I would on these initial parts. What one can do is make approximations similar to this. And this is known as one of the ways of making approximations called the dynamic mode decomposition. So a clever way numerically of computing these approximations of the matrix A. And we'd worked on how to do it very fast and in a streaming fashion. So if I give you online data, which is of a dynamical system, you can go back and also compute the approximations. So I'm going to skip some of this, which is details which are telling you about how you can do this efficiently numerically. And the onus is, of course, when you're doing this in a learning setting and doing this by batch renormalization and iterative sampling, you're getting into the fact that your training data, you can actually minimize this objective with the training data loss. But the real error you want to minimize is your generalization error, which is in time-dependent phenomena, your prediction error. So if you only get access to this data, can you make predictions and forecasts which are consistent with data you've already observed, and you can measure the loss? So this is showing for this sequence, the error is my predictions are from 8,000 timeframes to 10,000 timeframes. And of course, I'm normalizing them over that, but my true data set was 10,000. And so these error plots and all are really focusing on the generalization error of your learning. I'll skip this as well and just tell you how one can actually develop architectures. So, and deep learning architectures. So what you can do is go back and learn this A operator very easily if that's all you wanted to do, by either doing MLPs or even simple, if you wanted to do them in kernelized space using res nets, residual nets. So they all go into the architecture of these units that you're trying to learn. So what is being learned? What is being learned is to take your nonlinear data and embed it into a space where it's a linear. So you're learning the state space representations of your dynamical motion, both space and time. Now, if it was a simulation like I was showing you, then it's the values at every grid point of your simulation. If it's molecular dynamics, let me skip this for a second, I'll show you some examples. Then it's the trajectories of every atom and the relationships of every atom that is being learned from data. You're learning from, say, 8,000 time steps, and then you're predicting in the remaining time steps. And so this motion that you're learning is how is for the current Hamiltonian, how is this molecule behaving? But what we want to do is a little bit more. We want to learn also the B and the U, not just the A. So what I have to do is, if you told me and gave me a lot of training with known control U, I can take that and bootstrap myself and learn where the U is unknown as well. And the way you do that, and you can also do this what we call probabilistically, using probabilistic state space models, is to use something called a deep variational Bayes filter. But all that is nothing more than saying, I can map my U of t to a latent state space, which is given by my Z of t. And the Z of t is a function of time. And the Z of t has components A, B, and also in a probabilistic setting, it has CT, which is given by noise, process noise that is attached. And I have to learn A and B and F and C, T, I can make an assumption of what WT is. One of the simple assumptions is to assume it's a Gaussian. It's always a lower bound of what the true noise is. But under this scenario, again, you've got nice stochastic optimal control algorithms that people have developed that you can use to learn. The crux of the matter is that this learning space is done by this variational autoencoder, and the latent space, learning is coupled to thinking of this as a function of time. So there's a signal filtering and optimal estimation of the signal at every time step that has to be, in some sense, you can think of it as filtered or learned from a filter representation. And that's why you can think of the ZT advancing the latent space dynamics is like learning the optimal Kalman filter in the case of a linear Gaussian approximation. Here comes the final crucial part of this entire exercise. So what we have to do is learn the dynamics such that it's controllable. And also, it's controllable with an unknown U, or unknown f of t, the bias. So what the agent is going to be something called an actor critic. And there are many variations of this actor critic. I'm just giving you the basic one. There's a soft actor critic and so on. But what the agent is going to do is going to learn a control, which is an action of the best additive control through a double network of learning. So it's got two networks. Again, my mouse has disappeared. So you can see FC is a network it's going to learn, and FA is a network. And in these cases, these networks are learning functions. And they could be complicated functions, like it could be just a gigantic matrix, or it could be a tensor function, or something expressible in that form. Since we have units in our network that are tensorial, we can express our entire function that we're learning as a cascade of tensor functions. Of course, we're also given the ability of nonlinear activation, so we can learn highly nonlinear functions using these networks. But the essential function is learned by always solving a constrained optimization problem. So the agent control is to learn the solution of this optimization problem. It's minimizing, the best move it can make so that it brings its loss function closer, or minimizes its loss function, which is bringing the molecule closer to the final configuration, which is given by the guide that in the game. The action is choosing, the agent is choosing the best action, controlled by the forward dynamics, constrained by the dynamical equation. And the critic receives the actor's best action and simultaneously learns a way to provide what is the best value for that move, the reward function. So the best Q function that would give it that kind of, for that state and that action, what is the best reward you can obtain. And so these actor critics, which was a nice model proposed in reinforcement learning, can be harnessed into simultaneously learning how to shape this molecule in the best possible way. So this figure is kind of capturing how the agent is learning once you have a way to build a simulator of your molecular dynamics, which is not requiring a supercomputer, so you can do what ifs with. So FA gets the state and the current state, and it makes an action. So you can see the bottom line feeds into F of A, which makes an action U of T, which goes into F of C. F of C is the critic, like the coach. It sees what action you make and says, aha, it has this value. This is the reward. Of course, if given the current state, FA can query FC to see what is the best value move it should make. And FC is not a trained evaluator because the state space is so large. So it's going to learn, from the current value and the next move that the actor takes, what an improved value function is. So both FA and FC are learning simultaneously. One is learning to make the best possible move based on what the coach or the critic is telling it, what the best value function moves are. And every time the actor moves, the critic is learning, the critic or the coach is learning, aha, now that I've got a new sample, I can go back and tell you what the better value moves that you should make. So here are some examples of this actor critic, and it's still not fully there, but in making our predictions. But if you go back here, we are starting to see that, yeah, over training, it very quickly converges because the initial state was not that very far away from the final state. But you can see over here, strands are still outside the guide, so it's not getting the perfect solution. And then the predictions are also getting better and better over time. But again, you can see that, no matter how many times as you're going through, the strands remain in multiple places outside. I'll skip this and try to conclude because of time, because time is nearly up. One essential part that I missed, there are lots of other details, by the way. I'm so sorry that I can't cover everything, but I wanted to give you a glimpse of what it takes. But molecules are highly flexible, and mathematically they are, a product of motion spaces, right down to the atomistic or even, the backbone of the protein changing and the side chain motions and so on. So sampling is key. And this is one of the problems that one has. And what you want to do is, if you're trying to solve any optimization problem, you want to always get good sampling of your space. And this is getting into the theory of what is called low discrepancy sampling, which is a problem that has been dear to my heart. And one continues to, oops, somehow I can't move forward because this thing. Okay, so in reinforcement learning, and also in many of these where the options are unknown to begin with, you're very much like what I call multi-arm bandit problems. You have an unknown distribution. You don't know which moves to make because the value function is unknown. And if you're doing this in a probabilistic setting, then you're trying to sample the space, but sample it with important sampling. And one of the nice techniques was developed by Thomson in 1933. And this is something that we are harnessing together with low discrepancy sampling of product space, which is a quasi-polynomial number of samples other than an exponential number of samples that are needed. Because to keep the sampling of your space down is absolutely essential. So again, my things seems to be stuck, but let me go here. There are many other problems, by the way. Optimizing packing and docking that one had looked at earlier, and now I'm revisiting it in the lens of machine learning, and specifically in this for dynamical systems. Model assembly, spherical prediction. But before I conclude, here's something that we had done several years ago, but now we are trying to repeat it by trying to learn this assembly by itself, rather than, of course, this is just a synthetic example of assembly. This is not how nature assembles it, but we, for any kind of trajectory, you can learn how things assemble. So I'll stop there with acknowledgements. I owe a lot of thanks to my senior collaborators. Many of these names are outside computer science, but you learn from them because learning the domain well, and I'm thankful to them. like Tim Baker and Joachim Frank and Andy McCammon and Art Olson and many others. They let me come to their poker table and play poker with them as a computer scientist or a computational applied mathematician. My current group members, I owe thanks to Ahmed Abdul Kader, Noah Beal, Conrad Lee, Ryan Farrell, Chase and Levi, Yiwan. Some of the stuff I showed you also was done with my past group members, because revisiting old stuff. So with that, I'll stop, take questions. Sorry I had to rush a little at the end, but hopefully there are more questions. And I'm always contactable if you want more details. Okay.
Thank you so much for an excellent talk. And if folks have questions, you can put them in the chat or unmute yourself. But maybe I'll just ask one that is really current interest. I'm sure you saw a week ago the AlphaFold results and using machine learning in the CASP competition for protein folding. Do you see any changes in this research area going forward in the next 5-10 years with advances in machine learning?
Yeah. So my group and I had studied AlphaFold over the summer, AlphaFold 1, and now they're AlphaFold 2. And one of the things that I think it's a great step forward, but they've done is they did both learning of, sequence to structure. That was the mapping, right? And so hence they looked at the homology of all sequences and then also homology of folds, trying to learn how to fold the thing correctly from lots and lots of past examples. So that's the fundamental. The approach we are taking is, and I also looked at AlphaFold 2, and the nice thing is that this is great stuff, and they've also made their code available. The approach we've been taking is more biophysical, which is saying, can I not only learn to fold like nature, but I want to develop the essential control function, which is knowing the essential moves it has to take at the right time as it dances towards its solution. And once I learn those, I can control it maybe better. I can fold it faster. And I can also substitute, different things to bias that fold so that I can accelerate it, right? So the goal is has been slightly different for our group. But of course, we leverage from what they have. So, as far as even initially, if I need to get more examples, I can use AlphaFold 2 to give me a fairly good example of a folded protein, because there are lots of structure, lots of sequences available for which there are no folds available, right? No 3D structural folds. And ours is both, the dynamics has to start with the fold. So the ability to leverage from them is immense. But the challenges, this is, I think, from my understanding, just the tip of the iceberg of where machine learning can help. Because there's so many other things before we can develop a vaccine or a proper, a lead compound that would cut into the, years of trials and training and testing and soft, biology that has to be done to make sure that this thing happens. So there's a full, underneath that iceberg, full range of other beautiful problems that are very harnessable by computational science and math that we should start leveraging from. So
that's great.
I tell my students, and a rule I learned, I can see further and better just because I've learned how to clamber over the shoulders of giants. not just out to stand on them. So we always have to learn from each other.
Thank you. So that leads into a question that Jorge Aponto Gomez asks. And have you worked directly with any vaccine producers? And has any of this gone into the real world of the antivirals out there?
Yeah, so, the answer is yes, but it's been indirect. So there's groups here in Texas who have contacted me. There's Merck, there's there's an offshoot of Pfizer. What they, what our work is at the computational tools development level, right? So and all of our software is available. So they can download them. Now, when they came to me recently, which was about a year ago now, for almost a year, saying, we saw what you had done with HIV spike. We want to model and this was the group that is the computational biology group out of this. And so we gave them our software, which was available, and we then told them how they could use it. So this is some of the tools in docking that we had developed. It's called F2dock, F3dock, and PF2fit, PF3fit were the older tools of how you take a molecule and fit it together. And so this is the kind of interaction we've had. But it's a huge pipeline for development. Yeah. So we as computational scientists, that's the kind of interaction we've had. But one can become very specific because, when it came to the structure of the SARS, the entry mechanism, right? The current vaccines are all mRNA based, which is post-infection, right? So what we had been working on was for preventing infection. We don't want the thing to signal the AC2 receptor interaction. So that's where our focus has been. But the tools are generic. I'm hoping they're using them well. I want that vaccine too.
Well, thank you. I'm glad that you're working on this for the good of the world. Very, very honorable. So thank you very much.
Thank you.
So that concludes the seminar. And again, Dr. Bajaj, thank you for speaking, and thank you to everyone who's here on Zoom or watching through YouTube.
Thanks all. Have a good holiday.